Memang senyap lagi ke?
Cikgu sedang buat consultation dengan Afika sekarang.
Oh, maksudnya
Um,
present je.
ya, kita present je.
Oh, okay. Oh, sorry. Okay, sorry. Okay. Alright. Okay. So for this question, the diploid number of chromosomes in humans is 46, and the diploid number of chromosomes in rice is 94. Oh, belum baca dah. Okay, baca dah.
Bismillahirrahmanirrahim. Allahumma aftah alaina hikmataka wa insya alaina min huzaini rahmatika ya arhamar rahimin. Wa bishri lisiri amri wa halul uqdatam min lisani yafqahu qawli. Wa sallallahu ala sayyidina Muhammad wa ala ali sayyidina Muhammad walhamdulillahi rabbil alamin.
Okay. So the diploid number of chromosomes in humans is 46, and diploid number in rice is 24. What does this indicate about diploid chromosome numbers? So plant species have a lower diploid number of chromosomes than animals. So we can say that this is wrong because it's a generalization. So from the statement, even though it did say that Homo sapiens is 46 and rice is 24, but we cannot generalize, at least we cannot generalize yang all plant species all plant species punya diploid number is 24 sebab it's probably just rice. Okay. And then B, members of a species have the same diploid number of chromosomes. That is probably true. Okay, I mean this is true, but that is probably really the answer. And then like for C, the evolutionary progress of species is determined by the diploid number of chromosomes. It's a true statement, but it's unrelated to the statement yang dibagi dekat atas tu, because from kalau we read the original statement, it does not state anywhere anything about evolution. And then for D, the complexity of the organisms is correlated to the diploid number of chromosomes. Also, D is a generalization because like the only information that we have is that Homo sapiens is 46 and rice is 24. And there is also proof that diploid number of chromosomes is not correlated to complexity of organisms yang dalam slide teacher tu. Like I think there are animals yang even though like far more simple than us, tapi they have more number of chromosomes. So like that's why out of like A, B, C, D, B is the answer. Okay, everyone okay? Alright. Okay. Alright, so the next question. The genetic determination of dogs' coats can be quite complex with many different genes acting at the same time. So the dominant allele gives brown tones, and the recessive allele results in red tones. And the color intensity is due to another gene. The dominant allele B gives dark color, whereas the recessive allele B results in a light color. So what would the genotype of a light brown dog? So I put there EEBB to that was my annotation lah. Like the probable genotype for a light brown dog would be capital E, small letter E, and small letter B, or both capitalized E and small letter B. So genotype of light brown dog produced from a cross between a dark brown dog, which probably can be capitalized E and B, or capitalized E, capitalized B, small letter B, and a light red dog, small letter E, B. All right, next. So okay, so like from the question, actually kalau tak buat Punnett square pun tak apa, because okay, we know that the probable answer is either A or D, can either A or D. But looking at the parents ni, dark brown dog and light red dog, there is no chance that the answer is A, because for there is no chance that the answer is A, because for the light brown dog to have apa tu both capital E, both dominant LLE, the one of the parent, no, both of the parents kena ada dominant LLE. But from the question, dia dah cakap dark brown dog and a light red dog. So because one of the parent is a light red dog, they prevent the light brown dog to have both dominant LLE. Okay, so like you can just timbuk from there lah. But if you go and do the Punnett square, next slide. Kalau you do the Punnett square, so tadi kan kita ada like two like the parent tu, dark brown dog tu kan boleh ada dua genotype kan, either dominant LLE or one of ataupun like heterozygous E. Okay. So okay, and then when we do the Punnett square tu, Punnett square for like both homozygous dominant and homozygous recessive, we'll get all of the offspring tu akan jadi heterozygous. Akan jadi heterozygous. Alright. So okay, so tak adalah jawapan yang kita nak tadi tu sebab kita nak cari light brown dog kan. Sebab yang kalau dalam ni kalau we do the homozygous, the crossbreed between like homozygous dominant and homozygous recessive, we'll only get semua tu heterozygous, which is kita akan dapat dark brown dog. So okay. And then okay, I just put like over here the hybrid cross ratios, so just like for you to remember. And then macam, because like there are certain questions yang you know, like if you have all this, you don't have to menyusahkan diri to do the Punnett square. So I'll just explain the dihybrid cross ratios ini je. So like if the crossbreed is between homozygous dominant and homozygous recessive, like the ones yang I buat ni, between the light red dengan dark brown dog ni, because like one is like homozygous recessive and another is homozygous dominant, right? So the result is all heterozygous. So for crossbreed between homozygous dominant and crossbreed dengan homozygous recessive, you get all heterozygous. And then for the ratio, okay, 9331, the famous ratio ni, this one only dapat for between heterozygous and heterozygous. And then for 11111, okay, yang ratio 1111 ni, yang bila when we do the hybrid cross, kita akan dapat the recombinants tu, this ratio. And this ratio only boleh dapat when there is a crossbreed between heterozygous dengan a homozygous, that is a homozygous dominant or recessive. And then the next ratio ni, the 3:1 ratio ni, you akan dapat bila it's a heterozygous crossbreed dengan okay, macam mana explain? Heterozygous crossbreed dengan dia kan ada dua gene ni kan. So like for example, EEBB apa ni, small letter EB ni, the homozygous recessive ni crossbreed dengan let's say that capital E dengan small letter E. So and then the BB. Okay, tak apa. We'll see this one in the next slide. Okay, next slide. Okay. Sekejap. Okay. So, apa tadi tu yang 3:1 ratio tu happens bila a homozygous homozygous crossbreed dengan hetero apa tu? Sorry. Hetero and homozygous genes. So macam okay, like for example, this one kan homozygous recessive dia crossbreed dengan the capital E and the small letter E, but with like homozygous dominant kat belakang. So like B capital B, yang tu you akan dapat 3-1. Okay, but this is not the example for 3-1. Okay, sambung. So tadi, okay, back to the question. Kan kita nak cari like dark brown, apa tu? The light brown dot kan. So kalau you nak do the Punnett square, because there is two probability, either you crossbreed dengan either you crossbreed dengan apa tu, like the capital letter E and B, or dengan the capital letter E, small letter E, and capital letter B and small letter B tu. Okay, so we've done the apa tu yang the capital E and B tu. So now when we tengok yang the cross hybrid dengan capital E dengan small letter E ni, di slide yang tadi tu. Lagi. Macam this one. Okay, waktu ni baru kita dapat the light brown dot punya genotype tu. Which is yang di circle tu. This one. So like in both Punnett squares yang kita dah buat tu, we can see that apa tu, capital E, capital B and small letter B tu tak ada pun, kita tak dapat pun that one. So A is not the answer and kita akan dapat D sebab ni the genotype yang actually received from the parents, if the parents is like dark dog or dark brown dog, macam itulah. So like this is Punnett squares tu you buat kalau you rasa macam you're not confident untuk tembak lah macam tu. But yeah. Okay, teacher. Faham ke?
Faham.
Alright. Okay, but remember the dihybrid cross punya ratio tu. That one will help a lot. Okay. This one. How about this one?
Okay, Dina.
Yeah.
Haziq.
Alright. Jadi so korang boleh baca dulu soalan ni. Sekejap.
So, the Chinese soft-shelled turtle, Pelodiscus sinensis, lives in saltwater marsh. The turtle can live underwater and out of water. These turtles have fully developed lungs and kidneys. However, many microvilli have been discovered in the mouth of P. sinensis. A study was undertaken to test the hypothesis that oxygen uptake and urea excretion can simultaneously occur in the mouth. Initial experiment involved collecting nitrogen excretion data from P. sinensis, the turtle units both in water and out of water. When in water, it allows water waste products to be washed out of its mouth. When out of water, it regularly dips its head into shallow water to wash its mouth. The table shows the mean rates of ammonia and urea excretion from the mouth and kidney over 6 days. So, korang boleh tengok data dia. So, from this data, we conclude that when we are comparing between excretion of nitrogen by the mouth when the turtle submerged in water, the ammonia difference is not that different, where it's 0.29 in water and 0.30 out of water. While for urea, it have a quite huge difference, where when it's submerged in water, it is 0.9, and when it's out of water, it is 1.56. And next is excretion of nitrogen by the kidney. Turtle submerged in water, it will be 0.63, and while it is turtle out of water, it is 0.54, not much different also. While for urea, there is also a huge difference where when the turtle submerged in water, the excretion of nitrogen is 0.07, and while turtle out of water, it is 0.73. Right. It was noted that during long periods out of water, turtle immediately move their mouth to take in water from a shallow source and then discharge it. Changes in the dissolved oxygen and the quantity of accumulated urea in the rainwater discharged by the turtle were monitored over time as shown in this graph. Next slide, please. All right, so we could see from this graph, there is head of water and head deep into water. So when the percentage of water, the saturation of water dissolved oxygen percentage, when it is head deep into water, it will be decreased, and while it is head out of water, it will increase. And over time, we could see the accumulated urea increase bit by bit. But we could conclude that from this graph, whenever the head deep into water, the concentration of oxygen will decrease. Next, in order to test whether a urea transporter was present in the mouth tissue of the turtles, fluoretine, a known inhibitor of membrane proteins that transport urea, was added to the water, in which a further set of turtles submerged their heads. The results of the treatment are shown. So we could imagine that there's two types of water, where one water is where fluoretine is added, while another water there is no fluoretine. So next slide. All right, so from this graph, it shows the mouth urea excretion rate per micromole urea per day. So for control in which there is no fluoretine in the water, the mouth urea excretion rate is high, where for the turtle in submerged in the water that contains fluoretine, the mouth urea excretion rate is very, very low. So this shows that fluoretine has inhibit the membrane protein of that transport urea in the mouth. Next, further research was conducted to determine where mRNA expression of urea transporter gene might be occurring in P. sinensis. G electrophoresis was used to analyze different tissue samples for mRNA activity. So for this electrophoresis, they are used to check on the expression of a urea transporter gene. And we could see that there is quite a mRNA expression for roof of the mouth, and there's a lot of mRNA expression at the tongue. This is because of the color or the mRNA expression cause the band to become more thicker. Next. So next for the question is, compare and contrast the changes in urea excretion in the mouth with the changes in urea excretion in the kidney when a turtle emerges from the water. So for this compare and contrast question, it is important for us to analyze the graph first, and then we will highlight the main point of the question. And the main point will be the changes in urea excretion in the mouth with the changes in urea excretion in the kidney when a turtle emerges from the water. And we could see from the first graph the table data, there is a huge difference between submerged in water and when turtle is out of water. Because this question is only about compare and contrast. So it is important for us to only give the data based on the graph and not make our own reasoning. And next, we should also point out some few data such as the both increased by 0. Sorry, Jima. Table from the table here. Alright, so we could also do some calculation, and maybe by doing that calculation, we call, for example, the difference between urea is the same, which is increased by 0.66 when turtle emerges from water. And next, the increase is higher in kidney. I'm not sure if we can do this kind of calculation, but I think it's better for us to just say like increase is higher proportionally higher in kidney. But for the calculation, I'm not sure, because for this calculation, we have to look at the table and then we do some ratio. And then from the ratio, we compare like the excretion of urea in the when it is submerged in water and from the kidney and the mouth. But for the question, remember, it is for urea and not ammonia. So we must compare the urea and not the ammonia one. So try to not be confused. And the one that we have to compare it with is the mouth and kidney. Alright, and lastly, urea excretion by mouth is greater than kidney out of water. So just a simple comparison where we could see when we look at the table, we could see that urea excretion by mouth out of water is higher compared to in kidney excretion, the right one. Total out of water 0.73 and 1.56 out of water in the mouth. All right, so the next question is quite simple, where it told us to describe the trends shown by the graph for dissolved oxygen in water discharge from the mouth. So look at the graph where there is up and low and up and low. The second graph. Aku rancang nak dua question and then tanya faham ke tak. Okay je. Alright. So we got the second graph. Alright. The question asks us, describe the trend shown by the graph for dissolved oxygen in water discharged from the mouth. So we need to check the graph and then describe it, where when the head deep into water, the oxygen decrease. But when the head out of water, the oxygen increase. So just describe, and we do not have to reason or give any explanation. As the next question asks us, suggest reason for this trends in dissolved oxygen. Before that, do any of you have question for question A and B? Or maybe you doesn't understand the comparison, the percentage of question A. I think you understand for now, but whenever you have the question, do not be shy to ask me question through the chat. Next, suggest reason for this trend in dissolved oxygen. So for the next question, we need to explain why there is the decrease and why there is the increase. So it's quite simple, where when the head is deep into the water, maybe we could explain when the head is out of water, there will be a respiration, so the oxygen concentration will increase. And if the oxygen deep in the water, when the head dip into the water, the oxygen absorbed from water or exchanged for urea when head dip in the water. So that's why there is a increase in the oxygen concentration decreases. And next, when the head dip in the water, even though the turtle have lung, the lung cannot be used with head in water. So there is no respiration or there is no oxygen intake when head is under the water. So that's why there is no increase. And next, where oxygen from water in mouth, where it means that it refers to when the head is deep into the water, is used for respiration. That's why the oxygen decreases. And lastly, oxygen from air dissolve in water when head out of water. Yes, this in the first question, it states these turtles have the first description of the question in the first table. The first table. Never mind. In the first table or the starting of the question, it has stated clearly that these turtles have fully developed lungs and kidneys. However, many microvilli. So it states there that these turtles have developed lungs. So this shows that this turtle breathe through lungs. So if the turtle doesn't have any gills, so they cannot take oxygen from the water. And yes. And for question C, deduce with a reason whether a urea transporter is present in mouth of P. sinensis. It is obvious that there is a urea transporter is present. This is because referring to the third graph, Diana, maybe aku boleh nak presentkan Diana untuk lagi memudahkan. Diana,
Sekejap.
turtle tiada gills, turtle only lung. Alright, so aku share screen. Alright, can you guys see the screen? Alright, good. Alright, so deduce with a reason whether a urea transporter is present in the mouth of P. sinensis. Even though the first description of the question state that a study was undertaken to test the hypothesis, it is only for hypothesis. And then even though it states here that a known inhibitor of membrane protein transport, it is to check whether there is transport member protein that transport urea or not. So they check it by using the fluoretin. So to prove by using this graph, it is proven that macam mana kalau orang nak tahu tak ada link lang sebab turtles reptiles kan, kebanyakan reptiles saksanya akan ada cakap siap. Alright, and then we could see that this question check on the mouth of urea excretion rate. So they check at the mouth and not the kidney. And then when they check at the mouth, they also check using the control and the fluoretin. Thus, when there is fluoretin, it is proven that there will be no urea excretion when protein available. Thus, proving that there is a membrane transport in the mouth of the turtle. So less urea excreted or lower rate of excretion when fluoretin was present. So all of these answers that are almost similar are acceptable, but never zero. It is almost zero, but never zero. here. It is stated here not 0. So we cannot, if we say the answer is 0, then our answer would be wrong. And lastly, outline the additional evidence provided by the gel electrophoresis shown above. So, the electrophoresis are used to check whether there any mRNA that express urea transporter gene. So we could see that there is an expression of mRNA at the roof of the mouth and in the tongue. Therefore, this shows that there is mRNA only in mouth and tongue and not inside the esophagus, intestine, and kidney or bladder. But because like, why will you excrete urea into your esophagus? Alright, and then there's band or lines indicate mRNA for expression of urea transporter gene. By the way, this question is related to the previous question where if there is a urea transporter present in the mouth. So we must give our answer based on the last question where if there is in the mouth. So we should not mention that there is esophagus in or there is the gene for or the protein membrane transport protein in the esophagus, intestine, or kidney. And next, the urea transporter gene is expressed in mouth or tongue and not expressed made in esophagus or intestine or kidneys or bladder. And lastly, mRNA of the urea transporter is higher in tongue, more in tongue lah. So for now, any question? For questions B2, C, and D? Anyone? Jima, mata Jima nak tanya soalan ke? Merana memang tak ada. mRNA memang tak ada dalam kidney. In this, for this question, it shows that there is no mRNA activity in the kidney. Thus, there is no mRNA. There is no urea transporter gene in the kidney. So we could not make like, alah, mestilah ada, takkanlah dalam kidney tak ada mRNA. So nak tak nak kena tengok question. Kita nak buat assumption kita sendiri or kita punya own. Macam hari tu saya baca artikel yang ada transporter dalam kidney, tak boleh buat macam tu. Dia kena tengok soalan. Kena tengok dia punya graph. Unless kalau dia state that there is in the kidney, so of course lah ada mRNA dalam kidney. Good question, Kis. Any question? Alright, if not, we may proceed to the next question. So, you guys can read first. Alright, gibberellin promotes both seed germination and plant growth. Researchers hypothesize that the gene GID1 in rice codes for the production of a cell receptor for gibberellin. The mutant variety GID11 for that gene leads to rice plant with a severe dwarf phenotype and infertile flower when homozygous recessive. It is suspected that homozygous recessive GID11 plants fail to degrade the protein SLR1, which when present inhibits the action of gibberellin. The graph shows that the action of gibberellin on the leaf and alpha amyloid activity of white type rice plants and their GID11 mutants. So both of the graph is one for the length of second leaf, and another one is for alpha amyloid activity. And when we look at the, so remember that GID11 refers to the homozygous recessive rice plant that contains the mutant variety. So the key point here is homozygous recessive, where if the both of the gene for the mutant are expressed, thus this case will happen where we could see that GID11 for the both graphs have less to zero expression or growth. While when we compare it to the white type rice plants, there is a high growth. Next. Maybe we could explain to the next question. State which variety of rice fails to respond to gibberellin treatment. So this is quite simple, where we only have to look at the graph, and we could see that the growth for GID11 variant is very low, in which it fails to respond to gibberellin treatment, because we knew that gibberellin treatment is the hormone that promotes the growth or the seed germination for rice plant or any plant. Next, the activity of alpha amylase was tested at successive concentration of gibberellin concentration that produced the greatest changes in alpha amylase activity in wild type rice plants. So we just look at the graph and compare alpha amylase activity and which line shows the greatest growth or the greatest activity of alpha amylase, where we could say that it's from 10 to the power -8 to 10 to the power -7 mole per dm cube concentration that shows the highest growth. Very simple. For now, any question? No. All right, thank you. Discuss the consequence of crossing GID11 heterozygous rice plants amongst themselves for food production. So first, we should explain using, maybe not using Punnett grid, because we know that showing Punnett grid, Punnett square is something that will not give us any marks. So, but still, if you want to put it to more understanding, you could make a Punnett square here. And then you could see that the ratio is 1 to 3 for seed that will produce homozygous recessive. So this is one mark where we could say first the consequence of crossing GID11. So this is the consequences of crossing. And next, the consequence of when there is the homozygous recessive, which no response or it will inhibit gibberellin, result in less germination. And when there is less germination, so we could explain the consequences for food production, where there will be less growth. When there is less growth, it will produce plants with fertile flower, and at the end, it will lower rice production. It is important for us to state the last question because it at the last answer, because the question asks us for food production. So we need to explain the consequence of crossing. This is for consequence of crossing, and this is the consequence towards our food production. All right. Before we proceed to the next question, any question? All right. Mana dapat GG tu dia state tadi? There is dia tak state yang ada GG tu, but like I said, this is we make the Punnett square to make us more understand the question or the answer. When we are doing, like, imagine we are doing at the exam, and then we could think of the GID11 heterozygous. So maybe we could use A for the gene for the mutant, or the gene for the GID, but maybe for a better understanding, we should use G for the gene of GID11. And the GID11 it is heterozygous, and yes. So itu lain aja. So when it's heterozygous, there will be one dominant and one recessive. All right, thank you, Jima. So before we proceed to the next question, we should look first at the graph. Apa ni? Sekejap ya guys. Oh, okay, never mind. All right. So, most rice varieties are intolerant to sustained submergence underwater and will usually die within a week. Researchers have hypothesized that the capacity to survive when submerged is related to the presence of three genes very close to each other on rice chromosome number 9. These genes were not named sub1A, sub1B, and sub1C. The photograph below of part of a gel shows relative amounts of messenger RNA produced from these three genes by the submerged submergence intolerant variety O. sativa japonica, and by the submergence tolerant variety O. sativa indica at different times of a submergence period, followed by a recovery period out of water. So we know by now that O. sativa japonica is non-submergence tolerance or submergence intolerant variety, and while O. sativa indica is a submergence tolerant. And then the submergence is staged here from 1 to 10 days, and recovery, which is out of water, 1 to 3 days also. There is R to show that the day of recovery. All right. So we move to the question. All right. So determine which gene produce the most mRNA on the first day of the submergence period for variety O. sativa japonica. So for O. sativa japonica, we look at what the hell? What? Oh, faham dah. Alright, never mind. Alright, so which day is the band is most clearer? It will be in 1 and 2 and 1 and 1, 2, 3 and so it asks us for which mRNA. So the answer is sub1C. And next, the next question. So the next question is quite hard, where we need to outline the difference in mRNA production for the three genes during the submergence period for variety O. sativa indica. So we need to highlight O. sativa indica and not the O. sativa japonica, because the question asks us for O. sativa indica, where for sub1A is expressed strongly the most. We call outline, where sub1A have the highest expression, and while for sub1B it have the lowest expression because of the band. And next, sub1A express or produce mRNA for the longest time from day 1 to day 10, while for sub1B and sub1C, when it is submerged, there is not until 10 days of expression. And lastly, we could also state when we are different, so we shall always make difference in the strongest, the lowest, the longest, and the shortest. So for sub1A is the longest day, and sub1C is the shortest day. And lastly, using only this data, deduce which gene confers submersion resistance to rice plant. So we should think of what genes that give or grant the submersion resistance. That will be sub1A. Okay, maybe I would like to ask question. This is quite a simple question. Okay. Fatini, will you explain why sub 1A is the gene that confers submission resistance? Fatini?
Explain.
Yeah, just yes.
What was the question again? Sorry.
Why sub 1A is the gene that confers the submission resistance, or in other words, give submission resistance.
Because the DNA bands seen in sub 1A is more darker than sub 1B and sub 1C.
Wrong answer. Try to think.
Hmm. Pengek. Saya nak minta tolong.
Tak apa lah kalau begitu. So, Hamra, why sub 1A is the gene?
Tak tahu.
It's okay. So, why sub 1A is the answer? Because it stated here it is only expressed in indica sub 1B, while sub 1B and sub 1C are expressed in both rice varieties. Because look back at the question, where it stated here already that O sativa japonica is submerged intolerant, and while O sativa indica is submergence tolerant. This shows that there is something that O sativa indica have that O sativa japonica doesn't have, in which what gene there will be sub 1A that contains in O sativa indica but not in japonica. So it is the answer for sub 1A, and this shows that indica is the variety showing submission tolerance because it only shows in O sativa indica and not in japonica. Any questions so far? So what tips that I could give for this question is that read the question and also the data that they give at the beginning of the question. Any question?
No, but should we have a 5-minute break? It's 8:50.
Of course, yeah. Okay, so no question asked, so we could stop here.
By the way,
Thank you, Hazil.
just wanna say this, next time, untuk people yang present database question, two things. Number one, when you're presenting the basis of the database question, as in the situation, the long description, make sure explain it supaya us faham what's actually happening in the experiment. Maknanya don't just read it to us, tapi terang kat kami what's actually happening in the experiment. Sebab isu terbesar dalam database experiment is that kau baca eksperimen tu, lepas tu kau macam, apa benda kau buat ni? Faham tak? You don't understand what's happening in the experiment. So that's what's stopping us from being able to score well in the experiment. Number one. Number two, when presenting the questions and answers for each question, explain to us what the question actually want. Contohnya soal benda-benda macam discuss, evaluate, apa benda yang dorang nak actually, and where can we find the data. Itu je, sekian. Okay dah.
Yeah, yeah. So we will continue at 9 with Arham presenting.
So
Okay, so for database question, the last one. Figure 10 shows the result of an experiment in which seedlings of Sitka spruce, Acacia shechensis, were grown for 6 months in sterilized soil, either with or without fungi added. C was the control with no fungi added. The species of fungi added were Lacacia lacata, Matu lacatia, Lacaria amestia, Telophora terrestris from a tree nursery, Telophora terrestris from a forest, V or 5 Bacillus involutus, 6 Isolithus tinctorius. Okay, so that's the graph. Let's see what this graph is all about. The shoot dry mass and the root dry mass. Okay. So spruce ni macam pokok coniferous. So actually kalau kita tak tahu pokok apa pun kita boleh jawab sebab seedlings kan. Sebab overall how we know how seedlings grow, shoot first and then root, and that's all about it. So discuss the effects of the 5 species of fungi on the growth of the roots and shoots of the tree seedlings. So in this question, you ask what is the effects of the fungi. So it does not specifically state anyone or anyone lah. So specific anyone fungi. So we just need to give our view or our what we call our review on the graph, based on the graph for the 5 species, how it affects on the roots and shoots. So 4 marks. From what I can see, we can tell about okay, first of all kita boleh bagi tahu the greatest and the lowest. Okay, that's one. Actually that's 2 lah kan. That's 2 sebab ni 4 marks. And then we can also tell how everyone has significant effect, because everyone has effect. Because number 6 too don't have that significant of an effect. So I have 3 marks now. So maybe another one we can say that we can compare between the shoot and the root. Which one will have a greater mass? Which one will have greater effect? Which one that the fungi have affected most? So 1A, that's what it is asking. So repeat, we can tell about the greatest, the least, yang affect both side, and then kita tengok which part of the graph yang affected most, root ke shoot, and then we can see how every single one of them have a significant effect. Okay, addition of the fungus has an effect on both shoot dry mass and root dry mass. Both are the effects. Easy one. By the way, C tu kita tak boleh ambil jadi contoh ke apa-apa sebab C kan control. And then greatest, if we can see here, is the 5. Macam salah ni. Oh tak, dia meletak macam tu. Okay, so 5 Bacillus involutus. So we state the 5 or Bacillus involutus is the greatest overall root and shoot dry mass increase to the seedlings, to the tree seedlings. Okay, and then the least the number 6 kan, number 6 Bacillus tinctorius. So it has the least effect on shoot and dry mass. Okay, itu dah 3 marks. And then another one tadi, kita bagi tahu how the effect on shoot dry mass is greater than that to the root dry mass in overall of the tree seedlings. Okay, dah 4 marks. So discuss ni, sekarang ni kita akan tengok graph kita. Basically least or greatest, then we look generally, okay, flow dia generally lowest, greatest, and then kalau ada satu lagi kita letak je apa-apa yang kita rasa boleh dapat markah. Macam tu lah, at least that's in my mind lah. And then explain the effects of the fungi on the growth of the tree seedlings. Okay, this is a bit of syllabus going on here because we don't know much about it. But actually, if we think about it, how can a fungi affect the growth of a tree, of the seedlings, of tree seedlings? So actually, fungi can cause the roots or maybe the shoot to grow more, because in the roots it can cause more, it can become the other part, or what we call the increasing part of that root, macam struktur tambahan lah untuk root tu. So kalau root, kita boleh bayangkan, okay, kita bayangkan root, and then the root has another part of it yang macam kita ada root dia macam ada benda lain lah. So root tu dia akan accept more water, dia boleh accept more mineral. So it can also cause the root to grow more lah. That's the function of, or the effect of the fungi. Let's see. It can increase the surface area of the roots, allowing greater mineral absorption and greater water absorption, promoting plant growth. This is
how does it increase surface area actually?
Dia macam dia jadi apa dah? Symbiote punya apa?
Autotroph ke?
Bukan, commensalism lah benda nama dia. Ingat tak?
Symbiosis.
Symbiosis. Dia boleh dia in that type of relationship lah. So dia general knowledge lah ni, sebab dia tak ada dalam syllabus kita kan.
Faham.
Okay. So state the relationship between root growth and shoot growth in the tree seedlings. Ni kita boleh nampak sendiri kan. Sama ada kita nak buat the higher the shoot dry mass, the higher the root dry mass, terbalik. The higher root dry mass, the higher root growth, the higher the shoot growth, ataupun kita buat as the root growth increase, the shoot growth increase. So simple, or we can say the root growth and the shoot growth is directly proportional. Okay, so suggest a reason for the relationship. We can ni pun lagi satu about our knowledge lah. So before I see the answer, maybe anyone would like to try? Why do you think that the root can have greater effect? Why when the root grow, they should also grow? Why is it? If no one wants to answer, maybe I would like to ask Arham. Maybe because the root can tampung more of the shoot. Okay, that's a good answer. Anyone else would like to try? Okay, no one. Okay, so let's see the answer. B, that's correct. That's kind of correct, I guess. To B, more roots can support greater shoot mass. That is kind of correct. So if I were the examiner, I would like to give 1 mark for that. So this is all about what we call general knowledge. There's 1B and 2B, so 3 marks lah. Tak apa, kalau salah tak apa, masih banyak lagi markah lain. Using the data in figure 10, deduce whether the effects of closely related fungi on tree growth are the same. So closely related fungi. So how to know them? Mestilah yang nama sama. Nama sama kat sini kita nampak 1 and 2, and also 3 and 4. using the data in figure 10. Let's see the data 1 and 2. Okay, 1 and 2. 1 lagi besar dari 2. And we look at 3 and 4. 3 and 4. Oh, lagi besar dari 1 and 2. So we can say that 3 and 4. Salah baca. Oh, itu 3 and 4 yang sebelah sana tu. Okay. Tak nampak sangat apa ni kan. So I would say Ayib, what would you like to say on this?
On question C. Right, deduce whether the effects of closely related fungi on the tree growth are the same, and I think it would not be using figure 10. I think it would not be the same, because for every different species, the length of both the shoot and the root are different. And even for 3 and 4, which both of them come from the same species, they have different growth for shoot and root. So it's different for every single case.
So I think that could be an answer, because we are taking it not from past year, but let's see the answer. The 2 species of Lacaria and the 2 species of Tylophora all have significant effect. Conclusion is supported by Tylophora less, less so by Lacaria. Let's see what this means. So if we can understand the answer, it's actually because the 3 and 4 is a lot closer than 1:2. Can paham tak? Just about 3
or
dengan
closely
4.
related or closely related species.
Something like that. Okay. So we kena tengok yang mana yang lagi dekat. 3 and 4 is a lot closer, the readings, than that to 1 and 2. So they are significant effect. Okay, kita faham. And the theory is supported in 3 and 4, Tylophora, but less supported in Lacaria. So that is how we would like to answer the deduce weathered effects. So let's see back, discuss. We give the least, the greatest, the overall, and maybe the trend, if it's a trend graph. And then we have to explain about the graph, and that's a bit of general knowledge, and then relationship, and then reason for that relationship, and then deduce whether the effect. Okay, I think this is a lot more easier than Hazik's question, but I think we can still manage to answer this question. Okay, thank you.
Afika.
Hello. So next we move on to the part 2 of paper 2, which is factual recall. Okay, so for the first question, we are asked to identify the stage of mitosis labeled in the image and give the reason. So basically, when we see this question, we want to consider, or we want to note the chromosomes, the state of chromosomes. So from the label X tu, kita boleh tahu yang dia adalah telophase, ataupun late interface. Sebab apa? Sebab kita boleh nampak yang chromosomes tu dah start divided. So itu indicates the stages of telophase lah. So ini boleh difahami lah. Okay, so the next question. Okay, so then we are asked to outline what indicated by mitotic index taken from a tumor kan. So first kita perlu tahu apa itu mitotic index. Jadi mitotic index ni adalah indeks yang menunjukkan kadar mitosis ataupun kadar sel yang tengah melalui cell division. So kemudian kita perlu tahu yang bila kita analyse sel tisu yang diambil daripada tumor ni kita tahu properties dia adalah dia constantly divide kan. Dia sangat cepat dia punya division berlaku. Jadi mestilah mitotic index dia tinggi. So okay, itu soalan kedua. And then
Aziz, when the question says outline, apa yang sebenarnya dia nak daripada kita?
I think when this question says outline, dia nakkan kita cakap apa, maybe what happens and what are the reasons, kan? So basically, korang kena cakap yang higher mitotic index, but why? Kan?
Why itu bukan explain ke?
Oh, okay. So outline just beritahu Zima? Oh, okay. Faham. Okay, so next, jap. Soalan ni dah tak ada apa-apa soalan. Okay, tak apa. Next, DNA has region that does not code for proteins. So what are the function of this region? So basically, non-coding part of DNA ni ada banyak type lah. Ada telomeres, ada intron, ada non-coding RNA genes. Tapi so it's up to us untuk nak answer mana yang paling ingat. Tapi kalau biasanya lah benda yang paling kita akan ingat adalah first adalah telomeres lah, which is the regions of repetitive DNA at the end of the chromosome. So the function of the telomeres is to protect against chromosome deterioration bila cell to undergo replicate lah, cell to divide. And then itu first telomeres kan. Kemudian yang tertulis pada slide tu, promoters or operators, ini merujuk pada gene regulatory sequences mungkinlah. Okay, yang membantu dalam proses of transcription of the gene of the DNA itself. So itu je lah. Dan ada juga apa lain. Siapa ada jawapan yang lain yang nak, siapa nak utarakan jawapan yang lain? Hello. Okay, kalau tak ada kita pergi pada next question lah. Okay, so untuk ni DNA methylation, okay, boleh baca dulu.
Okay, for this question, this is a continuation question from the previous question. So, okay, kita baca sama-sama. DNA methylation has a critical role in gene regulation by affecting transcription. Samples were taken from 2 colon cancer tumors, T1 and T2, and 2 normal colon samples, N1 and N2. Okay. A particular gene was implicated as a possible cause of cancer. The promoter of this gene was cloned AJ. The data show the DNA methylation patterns from this sample. The numbers 32 until 269 represent different markers in the promoter. Okay, basically kat sini dia ambil 2 colon cancer, 2 different colon. First is cancer colon and then normal colon samples. So okay, T1 and T2 is the colon cancer colon sample, and N1 and N2 normal colon sample. So as you can see here, the black dot is methylation, the white dot is no methylation. So we can see here the difference T1 and T2 ada banyak dark circle kan. So compared to N1 and N1 dengan N2, meaning that colon cancer ni occurrence of methylation ni lagi banyak, lagi more frequent than normal colon sample. Okay, so tu pasal figure ni. And then first question, outline the difference in methylation pattern between tumorous and normal tissue samples. So the what I have already mentioned earlier, kita kena observe betul-betul graph ni. As we can see, there is much more methylation in the colon tumor samples than normal. Tumor and normal samples, the markers 258 and 269, similar degree of methylation. So sample markers daripada 32 sampai 210 ni ada a big difference in the frequency of methylation that happen between these 2 colon samples. Tapi for markers 210 until 269, 258 dengan 269 ni je, we can see that there is no huge difference between these 2 kinds of samples. So kita boleh juga letak that observation in this question as the answer. Okay. Second question, suggest a way methylation may affect tumor cell genes. So okay, after this kita akan recall balik what is methylation. Tapi as for the answer for this question, DNA methylation may inhibit transcription of genes that would prevent cancer formation. And then, or the answer could be DNA methylation may increase mitosis or cell division leading to tumor formation. So, okay, basically methylation is part of transcription regulation. Yang kita ingat ada methylation dengan acetylation kan. So methylation ni, it basically maintains the positivity of histone of the DNA. So DNA kan negative kan. So when there is negative dengan positive, meaning that they attract, so that the DNA is more compact. DNA is more compact. So bila DNA more compact, dia tak boleh undergo transcription, which the DNA being transcribed into mRNA. So dia tak boleh, dia macam transcription tu akan berkurang lah if methylation happen. Sebab attraction between histone dengan DNA tu. Okay, next. Next slide. And this is the recall part. Okay. Sorry, types of modification. Ada acetylation, methylation. So acetylation ni dia neutralizes positively charged histone. Okay. Acetylation ni dia neutralize positively charged histone, allowing less condensed structure with higher level of transcription. So we can see here at the right diagram, we can see acetylation. So the DNA is very loosely packed. So the DNA is more open for transcription. Whereas we can see for methylation, it maintains the positive charge, making DNA more coiled, and reducing transcription. So the DNA is more coiled around the histone tail. You can see, so for the DNA to DNA kan double stranded, and then in order for transcription to happen, it needs to separate from one another kan, so that the antisense strand can transcribe into mRNA. But if there is methylation, that cannot happen sebab DNA tu tightly packed together. Okay, that is all from me. Next slide.
Okay, next slide. The question is, in the pea plant, Pisum sativum, the allele for tall plants is A, and the allele for short plants is small a. The allele for green plants is big B, and then the allele for yellow plants is B. Determine the phenotype of big A, small B, small b. So big A, small a, we know it's going to be tall, and then small b, because both are recessive, we know it will be yellow. Easy question, right? So we can just move on. Okay, compare the information that could be deduced when the genotypes are presented as small AABB or AB with the line AB like that. So compare, you have to remember, you have to put the similarity and differences. Only if visit contrast or state the differences, baru you letak differences only. But if compare, make sure to remember to put similarities and differences. So the similarity between these two allele is that both of them are allele for tall and green. However, the differences is that the one without the line indicates unlinked gene, and the one with the line indicates linked genes. So we just to remember, linked genes are genes that have the same gene on the same chromosome. So the one without the line are not on the same chromosome, the genes. Okay, next.
Kejap. Tak silap waktu awal tahun dulu, teacher ada cakap yang new syllabus ni bila dia cakap compare and contrast, only then similarities and differences. Tapi tak sure lah kalau compare.
True.
Syllabus sama kan?
Tapi because nowadays, right, they use compare and contrast. Then you have to come up with both similarities and also differences. Tapi kalau katalah, right, they use again the term compare. So memang kena beri similarities and differences also. Is that clear?
Okay, teacher. Okay, next. Okay, the question is, deduce one possible recombinant offspring of individual small big A, big B, and small a, small b after a test cross. So we have to know what is a test cross first. Just to remind everyone, a test cross is the cross of an organism with unknown dominant genotype with an organism that is homozygous recessive. So basically we have
Kejap, wait, Ali. Again, the cross of an organism with unknown dominant genotype with unknown dominant phenotype, supposedly.
Oh, okay, sorry.
Right? Betul tak? Then only becomes unknown. So the organism shows dominant phenotype. Okay. And we do not know whether the organism is homozygous dominant or heterozygous individual.
Oh, because saya dapat definition ni online. Salah kot, sorry teacher.
Ya ke? Unknown dominant genotype. Oh, mungkin betul. Mungkin di sini dia masukkan the cross of an organism with unknown dominant. Dominant tu refer kepada phenotype dia. Maksudnya unknown genotype of a dominant phenotype. And then dia simplify it macam ni. You faham macam mana? This sentence? Respond please.
Saya faham dia guna that genotype dengan homozygous recessive. So dia macam mana? Macam buat test cross biasa and then dia tanya like apa recombinants yang possible. Macam tu Kak teacher.
Tak. Yang the sentence from test cross tu. Definition of test cross. The cross of an organism with unknown dominant genotype. Alright. So actually it refers to unknown genotype, right? Unknown genotype of an organism that shows dominant phenotype. Is that what you understand from this sentence?
I think first glance dia tak nampak macam tu, but like after teacher explain, then it's become more clear, I think.
Earlier, Dina faham macam mana?
Kalau earlier pun, I think I would thought yang dia cakap pasal unknown phenotype but with dominant genotype.
Unknown?
I would think yang ni lah, dia ada like dominant phenotype but with unknown genotype. But I think like the wording macam pelik sikit. It's confusing.
Yeah, confusing ni, this word ni. Alright, so be very careful here. So actually this sentence is referring dominant to actually refer to the phenotype. Alright, so unknown genotype of an organism that shows dominant phenotype. Okay, with an organism that is homozygous recessive. And then only becomes clear. So hati-hati juga ya. Tapi kalau faham tak apa. Cuba takut salah faham. Okay, Ali teruskan.
Okay, next slide. So this is how I figure it out. So first kita pecahkan the gametes. The first one is big A, big B, and then a line, and then small a, small b, and then a line. And then we have to also separate it into recombinant genotypes juga. Big A, small b, a line, and then small a, small b, a line. The reason why kita ada recombinant genotype, kalau tengok dekat study guide, diorang ada crossing over, so there will always be recombinant genotype. And then because of the crossing over, it produces a recombinant genotype, and then kita buat test cross. The test cross needs to be both recessive, so small b, small a, small b, and then you times it, and then you combine it, and then dia akan jadi 4 ni, which is big A, big B, small a, small b, small a, big small b, and then big A, small b, small b, and then small a, big B, small b. So kita dapat dua allele yang sama dengan parents dia, and then kita dapat dua recombinant genotype. So kalau pergi balik, kita nak recombinants, right? So recombinants kita nak yang tak sama dengan the parents punya genotype. So it's either big A, small a dengan small b, ataupun small a dengan big B, small b. Sebab dia lain dengan the parents punya allele. Tapi kalau nak rasanya lagi baik if you do the yes, or you can just remember the ratio. Kalau anything that has to do with test cross is usually 1:1:1. If you see the next slide pun sama. you will get the result to be 111. Dia punya phenotype. Yeah. So
phenotype is 1111 provided that there is crossing over, yeah.
that's it for this question.
Faham ke semua orang ni? If let's say without crossing over for linked genes, test cross. Berapa ratio dia? Anyone? Cikgu sebenarnya suspend ni. Cikgu bersama you all ke cikgu tak bersama you all ni?
Tak hafal, teacher.
Ada, teacher.
Oh, ada, itulah diam saja. Cikgu takut cikgu dah terkeluar. Okey,
Hafal,
tak
teacher.
ingat.
Ada tapi tak ingat. So,
One lah. If linked genes, the hybrid linked genes kan dia behave macam monohybrid. If like say there is no crossing over. Tak ingat ya. Okay, tak apa. Take your time. Alright, baca balik genetik. Genetik ni kalau dah nampak semua senang saja. Alright. So this one is linked genes with crossing over ni ya. If you look at the question tadi, once they say there is recombinant, tengok balik soalan tadi. Right, so you can see from the question, deduce one possible recombinant offspring of individual, and then notation used is for linked genes, right? After a test cross, automatically we know that there must be crossing over. Betul tak?
Teacher.
Ya. Sekejap. Ni maksudnya mesti ada crossing over sama here. Okay. So kita akan dapat lah, kalau earlier kita dapat, without crossing over kita dapat ni 1, and then 1, the other one is this, right? So 2 saja. And then test cross, kalau test cross mestilah cross with homozygote recessive. Betul tak?
Yes, teacher.
Yes.
So dapatlah nanti, sini homozygote recessive. Dia sama je kan? Betul tak? Sama. So ni pun samalah. So kita akan dapat ni dengan ni dengan ni. So ratio will be 1 to 1. Alright. But then when it says recombinant, automatically we know that there must be crossing over. Because this is linked genes, crossing over. So instead of dapat 2 yang asal ni, kita akan dapat ni 1, 2, and then crossing over kita akan dapat another 2, right? Another 2. Satu akan dapat capital A and small b, and the other one is small a, capital B. Betul?
Yes, teacher.
Right, so dapatlah 4 from this. So 4 cross dengan yang test cross still samalah, yang macam ni. Alright, one type, the same type only. So resulting in ratio 1 to 1 to 1 to 1. Betul. So mana-mana yang ada baru, new combination ni will be the recombinant. Okay. Okay, very good. Lagi? Dah habis masa ya? Cikgu untuk perasan ni.
Ya, teacher.
Oh yes. Alright, seberapa soalan tadi dapat cover?
We finish all database question, and then untuk factual recall siap berapa yang tadi? 4 3? Few jugalah.
Oh, okay, alhamdulillah. Alright, so cikgu miss dapat lah database. Okay, anyway, very good lah. Alright, so the rest, tadi siapa yang present? Besides Ellie? Ellie Fatini tadi ya?
Haziq and Arham.
Haziq and Arham. Okay, okay. Thank you so much. Alright, so next class we continue lagi lah. Okay, Okay, so see you next week insyaAllah. So ada lagi kan? Next week ada lagi lah. Database boleh ke masuk fact out of our syllabus? Fact patutnya tak boleh lah. Tapi if let's say you apply the application of the content, can. Okay, tapi kalau fact tu not in our syllabus, patutnya dia tak. Okay, but sometimes it is different kalau if let's say it is general knowledge. So kalau soalan kalau tanya pasal suggestion usually you have to also relate with your general knowledge. Okay, Azimah?
Okay, teacher.
Okay, alright. So thank you so much. So let's end this session with doa.
Bismillah. Astaghfirullahaladzim. Subhanakallahumma hamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu ilaik. Bismillahirrahmanirrahim. Wal 'asri innal insana lafi khusr illa allazina amanu wa amilus solihati wa tawasaw bis sabr.
Amin, 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 Rabbil alamin. Alright, bye. Assalamualaikum. Thank